Okay, so we are up to the last lecture on the nervous system. I know the nervous system is a really big topic, that's lots to cover, but you've nearly made it through, and hopefully you are starting to see how some of all this content actually comes together. So this last lecture will cover the autonomic nervous system, and we actually talked a little bit about the autonomic nervous system right back at the start in our nervous tissue lecture. So I promise that this will be a much shorter lecture than the last two. We only have 4 learning objectives to cover. So we'll do a little bit of a refresher on the differences between the somatic and the autonomic divisions of the nervous system. We'll compare the function and the structure of the different divisions of the autonomic nervous system. And we will take a look at the neurotransmitters used by these two different systems. So learning objective number 1 is to compare the somatic and the autonomic divisions of the peripheral nervous system. So this image here we've seen before. It is describing the organisation of the nervous system. So we have our central nervous system, which is our brain and our spinal cord up the top here in yellow. We have our peripheral nervous system, which is everything outside of our brain and spinal cord, down here in purple. We then divide our peripheral system into 3 different divisions. So we have the somatic nervous system, we have our autonomic nervous system, and we have our enteric nervous system. Now, for this learning objective, we're going to mainly focus on our somatic and our autonomic nervous systems, mainly because the enteric nervous system relates to the nervous tissue of the gastrointestinal tract, and we don't actually cover much of that in this unit. If you go on to do systemic anatomy and physiology, you will cover all of this information there. So starting with our somatic nervous system, and this division includes both sensory and motor neurons. So our sensory neurons, which transmit information towards the central nervous system, they transmit this sensory information from receptors for both somatic senses, so things like our touch, pain, proprioception, as well as our special senses. So our special senses are sight, hearing, taste, smell, and equilibrium. All of these sensations are consciously perceived by the brain, meaning that we are aware of them. So we can feel when someone touches on our skin. We know when we hear something or we see something. The motor neurons of the somatic nervous system, so when the motor information comes down from our central nervous system out to our peripheral nervous system, these motor neurons innervate our skeletal muscle. So we're going to look at the muscular system next, but our skeletal muscle is the muscle that we can voluntarily control. And these somatic motor neurons play a role in both reflexive movements as well as the muscle contraction that we determine. So you want to pick up a pen or go for a run. Looking at our autonomic nervous system, and this division also contains both sensory and motor neurons. The sensory neurons transmit sensory information from autonomic visceral sensory receptors. So these are mostly interoreceptors. So they're receptors within our body that are located in things like our blood vessels, our organs, our muscles, and their job is to monitor the internal environment of the body. So an example of an interoreceptor are the chemoreceptors that monitor the blood carbon dioxide levels within our blood. They could be mechanoreceptors that monitor the degree of stretch in the walls of, say, our blood vessels or our organs. And with these visceral sensations, we're usually not aware of them. So we're typically not aware of the changing levels of carbon dioxide in our blood. We're typically not aware of the stretching of our blood vessels or the stretching of our, say, stomach. Now, the motor neurons of our autonomic nervous system innervate effectors including cardiac muscle, smooth muscle, and our glands. So these are the effectors that we cannot control. The autonomic nervous system regulates activities like heart rate, like your breathing depth and rate, the digestion of our food, and the secretion of hormones. The autonomic nervous system regulates these activities by either increasing or decreasing innervation to these different organs. Now, if we compare the somatic and the autonomic nervous systems structurally, or compare their anatomy, there are a few differences. So this first neuron that we have up here is a somatic motor neuron. The three below are our autonomic motor neurons. So if we take a look first at our somatic motor neuron, so firstly, the axon of a myelinated somatic motor neuron extends all the way from the spinal cord to the effector, so to our skeletal muscle. In contrast, most of our autonomic neural pathways consist of two motor neurons. So say one here, one here. The first neuron in this pathway is called a preganglionic neuron, and it has its cell body somewhere within the central nervous system, so either up in the brainstem, up in the brain, or in the spinal cord. Its myelinated axon then extends out of the spinal cord, out through, say, a cranial or a spinal nerve, and to an autonomic ganglion. So remember, a ganglion is a collection of cell bodies in the peripheral nervous system. The second neuron in that pathway is called a postganglionic neuron because it's post that ganglion. And its unmyelinated axon extends from the ganglion to the effector. However, in some of our autonomic motor pathways, the first neuron doesn't extend to a ganglion. It extends to specialized cells that we call chromaffin cells. So that's these here. So our chromaffin cells are in the adrenal medulla, which is the inner portion of our adrenal gland, which makes up part of our endocrine system. And chromaffin cells secrete the neurotransmitters epinephrine and norepinephrine. So you may also know these neurotransmitters as adrenaline and noradrenaline. And through those neurotransmitters, that's how we communicate with the effector. In our somatic nervous system, all of our motor neurons will release the neurotransmitter acetylcholine. In the autonomic nervous system, so looking at these three neurons down here, they will either secrete acetylcholine or that epinephrine or norepinephrine. Now, another difference between the somatic and the autonomic divisions of the peripheral nervous system is that the autonomic nervous system can then be further divided into two divisions. So we have a parasympathetic division and we have a sympathetic division. And these two divisions is what we're going to focus on for the rest of the lecture. So most of the organs that the autonomic nervous system innervates are under both parasympathetic and sympathetic control. In many of the organs, nerve impulses from one division of the nervous system will increase activity in that organ, while nerve impulses from the other division will decrease or inhibit that organ's activity. As one example, nerve impulses from the sympathetic nervous system to our cardiac muscle will increase our heart rate. Nerve impulses from our parasympathetic system to our cardiac muscle will decrease our heart rate. For this reason, the sympathetic division of the autonomic nervous system is typically called our fight or flight division because it results in increased alertness and an increase in metabolic activities that prepare your body for an emergency. And then our parasympathetic division is often referred to as the rest and digest division because its activities restore energy, conserves energy, and occur during periods of rest and digestion. And so that is going to be the focus of learning objective number 2. So we're going to look at comparing the function of the two divisions of the autonomic nervous system. And so ANS here, autonomic nervous system. So as I mentioned earlier, most of the organs of the body receive innervation from both divisions of the autonomic nervous system. Typically, they will work in opposition to one another. So if the sympathetic nervous system does one thing, the parasympathetic nervous system will typically do the opposite. Now, during periods of physical or emotional stress, the sympathetic division of the autonomic nervous system will dominate over the parasympathetic division. What that means is that we will have high sympathetic tone or more nerve impulses coming from the sympathetic division. These nerve impulses support bodily functions that allow for vigorous physical activity and the rapid production of ATP or energy. At the same time, our sympathetic division also reduces the effects of activities that would conserve energy. Besides physical exertion, various emotions, so things like fear, rage, embarrassment, can actually stimulate the sympathetic nervous system. And often these activities are called E activities. So it's a nice way to remember things. Things like exercise, emergency, excitement, embarrassment. All of these scenarios or conditions is when our sympathetic nervous system is going to dominate. So activation of the sympathetic division of the autonomic nervous system also stimulates the release of a number of hormones from that adrenal medulla. And the release of these hormones set in place a number of physiological responses that relate to that fight or flight response. So this is things like dilation of the pupils in the eye so that you can take more information in visually. Increased heart rate, so the blood and the nutrients are pumping around your body. Dilation of the airways, so we can get more oxygen into our lungs and carbon dioxide out. A reduction of blood flow to non-critical organs like the kidneys and the gastrointestinal tract. So in the case of an emergency, let's say you're running away from the scene of a crime, hopefully you're not the perpetrator, we don't really care about our gastrointestinal tract digesting our food. We would rather the blood go to our muscles. And then we also have an increase in the breakdown of glycogen to glucose in the liver. So we store our glucose as glycogen in the liver, and then we break it down and put it back into our blood when we need to use it. So all of these examples are activities that would benefit you in an emergency or a fight or flight situation. In contrast to the fight or flight activities of the sympathetic division, the parasympathetic division of the autonomic nervous system enhances our rest and our digest activities. This description more recently has also been expanded to include our breed and our feed activities. So this relates to the things that occur after eating, like digestion, taking the nutrients into our blood, defecation or going to the toilet, as well as the activities that allow us to breed. So for example, the parasympathetic nervous system controls the male erection. Parasympathetic responses will support functions that conserve and restore body energy during periods of quiet rest and recovery. And at the same time, parasympathetic innervation will dominate sympathetic innervation to promote activities like allowing food to be digested and absorbed into the blood, and then reducing the function of activities that support physical activity. So for example, slowing down our heart rate, slowing down the blood flow to our muscles, reducing the conversion of glycogen into glucose. Our parasympathetic activities are often called our D activities. So we have digestion, which is taking in food, eating it, and the nutrients be absorbed into your blood. Defecation, which is going to the toilet for number 2. And diuresis, which is that process of filtering our blood to produce urine. Other activities that are controlled by our parasympathetic nervous system include the release of hormones and other secretions that assist the breakdown of food, constriction of blood vessels to redirect blood flow, the slowing of the heart rate, and an increase in activity in our urinary system. So we're filtering more of our blood and producing urine to excrete those waste products. All the activities that take place predominantly while we're resting. So now, having compared the functional differences of the sympathetic and the parasympathetic divisions of the autonomic nervous system, learning objective number 3 is to compare the structural differences. So we established earlier in the lecture that the motor pathways in the autonomic nervous system generally contain 2 neurons from the spinal cord to the effector. The first neuron in that pathway is called our preganglionic neuron because it becomes before our autonomic ganglion. It has its cell body in the brain or the spinal cord. Its axon extends out of the spinal cord through a cranial or a spinal nerve. And the axon of a preganglionic neuron is quite small in diameter and it's myelinated. So these messages propagate very quickly. In the autonomic ganglion is where the preganglionic neuron will synapse or meet and communicate with our postganglionic neuron. Our postganglionic neuron lies entirely outside of the central nervous system in the peripheral nervous system. Its cell body and dendrites lie within the autonomic ganglion. The axon of this postganglionic neuron is also small in diameter, but typically it is unmyelinated. From the ganglion, it will extend all the way to our effector. Now, looking specifically at our 2 divisions, so here we will first look at our sympathetic division of the autonomic nervous system. So the sympathetic preganglionic neurons have their cell bodies in the lateral grey horns, so the grey matter in our spinal cord, in the 12 thoracic lumbar segments of the spinal cord, and then the first 2 to 3 lumbar segments of our spinal cord. So not all the way down our spinal cord, just in that thoracic region and the top of our lumbar region. In our sympathetic division, the ganglion, so where our pre and post ganglionic neurons will synapse or meet, is found really close to the vertebral column. So the ganglia in pathways that innervate the effectors above the diaphragm. So the diaphragm is that muscle that sits below your lungs and helps you breathe. So everything diaphragm up, so think head, neck, shoulders, and heart. The ganglion for those effectors are called our trunk ganglion or the vertebral chain ganglion. So they lie in a vertical row either side of the vertebral column. So you can see that here and you can see that here. These ganglia extend from the base of the skull all the way down to the coccyx, so the very bottom of that vertebral column. The other effectors, so the ganglia in the pathways that innervate our other effectors, so all the organs below the diaphragm, actually lie in front of the spinal cord. So you can't see it on this image here. I can show you another image on the next slide. And these are called the prevertebral or the collateral ganglia. So diaphragm up, we have our trunk ganglia on either side of the spinal cord. Diaphragm down, we have our prevertebral or our collateral ganglia, and that sits in front of the spinal cord. Because all of the ganglia in the sympathetic division of the autonomic nervous system are found quite close to the spinal cord, so either side or in front, the preganglionic sympathetic axons are quite short, but then our postganglionic sympathetic neurons are long. So it's only a short distance to go from the spinal cord to the ganglion, But it's a much larger difference to then go from the ganglion to our effector. Once a preganglionic sympathetic neuron passes through the spinal cord, it can synapse with the postganglionic neuron in a number of different ways. So either first, it can exit the spinal cord and directly synapse with the first ganglia that it reaches, synapse there with our postganglionic neuron. It can exit the spinal cord, reach the first ganglia, and then either ascend or descend to another ganglia, where it synapses with a postganglionic neuron there. It can leave the spinal cord, go through this trunk ganglia, turn around and come back to our prevertebral ganglia, or the ganglia that's in front of the spinal cord, and there synapse with our postganglionic neuron. Or, which we don't have an image of here, it can continue directly through this trunk ganglia to the side, and then synapse with those chromaffin cells in the adrenal medulla. It's also important to note that a single sympathetic preganglionic neuron, so one single preganglionic neuron in the sympathetic division, can actually have many branches of the axon, meaning that it can synapse with up to 20 different postganglionic neurons. So one preganglionic neuron can synapse with up to 20 postganglionic neurons. This pattern of projection is called divergence, and it helps explain why many of our sympathetic responses can affect almost the entire body simultaneously. So as an example, when you get really nervous, perhaps you have a big presentation or you're about to give your first lecture and you just kind of feel sick a bit everywhere. It's because your sympathetic division of the autonomic nervous system will be most active. And because of this pattern of divergence, it can affect lots of different effectors within our body. So this image is just to give you a bit more of a close-up of the different ways that a preganglionic sympathetic neuron can synapse with a postganglionic neuron. So firstly, we had it going from the spinal cord, through the spinal nerve, to the ganglion, and then directly to the effector. It can leave the spinal cord, go to a ganglion, and then it can either ascend and go up, or it can descend and go down, and then synapse with that postganglionic neuron and go towards the effector. It can go from the spinal cord to our trunk ganglia on either side of the spinal cord, continue through that and then go to our prevertebral or collateral ganglia, which is the ganglia that's in front of the spinal cord. Or it can extend through the spinal cord, through the ganglia, and to our chromaffin cells. So here it doesn't synapse with a postganglionic neuron. The adrenal gland will release the epinephrine or the norepinephrine, and it will communicate with the effectors that way. So now taking a look at our parasympathetic division of the autonomic nervous system. And so the cell bodies of the preganglionic neurons of the parasympathetic division are located in the nuclei of 4 of our cranial nerves in the brainstem. And then in the lateral grey horn, so the grey matter within the spinal cord, in the 2nd to the 4th sacral segments of the spinal cord. So our sympathetic division was our thoracic and our lumbar vertebra. Our parasympathetic division are up here in our brainstem, and then down here in our sacral region. Unlike the sympathetic division, the parasympathetic division has its ganglia located either quite close to or actually within the wall of its effector. So it's not close to the vertebral column, it's closer to the effector. And these are called our terminal or our intramural ganglia. We've got terminal ganglia up here. Because these ganglia are located close to or within the effector, in our parasympathetic system, our preganglionic neurons are quite long and our postganglionic neurons are relatively short. So the opposite to our sympathetic division. Also, within our parasympathetic division, our preganglionic neurons typically only synapse with a handful of postsynaptic neurons, so up to 5 postsynaptic neurons. All of these then only supply a single effector, and this explains the much more localized effect of the parasympathetic division of the nervous system compared to our sympathetic division. And then our last learning objective, not only for this lecture, but also for the entire nervous system, we will compare the neurotransmitters of the two divisions of the autonomic nervous system. So a neurotransmitter is a chemical substance released by an axon terminal that diffuses across a synaptic cleft and imparts an impact or an effect on either another neuron or some kind of effector. Typically, our neurotransmitters will bind to ligand-gated ion channels on the surface of that postsynaptic cell, which then allows for the continuation of an action potential. Now, within the autonomic nervous system, neurons are classified based on the neurotransmitter that they produce and then release. These neurons are either cholinergic neurons, if they produce and release acetylcholine, or they're adrenergic neurons, if they release norepinephrine. Now, no need to write that down. We're going to cover all of that again and in more detail on the next few slides. So our cholinergic neurons release the neurotransmitter acetylcholine. So you'll see acetylcholine written as ACH. In the autonomic nervous system, cholinergic neurons include all of our sympathetic and all of our parasympathetic preganglionic neurons. So all preganglionic neurons are cholinergic. They include the sympathetic postganglionic neurons that innervate our sweat glands. And they include all parasympathetic postganglionic neurons. So to recap that, cholinergic neurons are all of our preganglionic neurons, all of our parasympathetic postganglionic neurons, and the sympathetic postganglionic neurons that innervate our sweat glands. Now, acetylcholine is stored in the synaptic vesicles of axon terminals, so those little holes or spaces within the axon terminals, and they're released by exocytosis. So we talked about exocytosis back in our cells lecture. It then diffuses across the synaptic cleft, which is that small space between the neurons or the neuron and the effector. And then they bind with specific cholinergic receptors in the postsynaptic plasma membrane, so the surface of that postsynaptic cell. The two types of cholinergic receptors, both of which bind acetylcholine, are nicotinic receptors and then muscarinic receptors. So we'll talk about that next. So our nicotinic receptors are found on both our sympathetic and our parasympathetic postganglionic neurons. They're found on the chromaffin cells in the adrenal medulla and on the surface of the muscle at the neuromuscular junction. So the neuromuscular junction is that synapse between a neuron and a muscle, and which we'll learn all about next when we talk about the muscular system. Nicotinic receptors are named nicotinic because nicotine, the chemical, mimics the action of acetylcholine binding to these receptors. When acetylcholine binds to a nicotinic receptor, it causes brief depolarization or brief excitation of that postsynaptic cell. So it's likely to pass on that action potential. Our other type of receptor is our muscarinic receptor. These are found in the plasma membranes or on the surface of all of our autonomic effectors that are innervated by our parasympathetic postganglionic neurons, as well as those sympathetically innervated sweat glands. Muscarinic receptors are named muscarinic because a mushroom poison called muscarine mimics the actions of acetylcholine binding to these receptors. Activation of our receptors by acetylcholine can also cause depolarization and therefore excitation of that postsynaptic cell, but sometimes they also cause hyperpolarization or that inhibition of the postsynaptic cell. So it stops that action potential moving on. And this just depends on the particular cell that those receptors are found on. As an example, binding of acetylcholine to muscarinic receptors inhibits or causes relaxation of smooth muscle sphincters in the gastrointestinal tract. So we can have food move through that process. Whereas binding of acetylcholine to muscarinic receptors in smooth muscle fibers around the iris of the eye causes contraction and it causes the muscle to contract. So we let less light into the eye. So it just depends what cell or what effector those receptors are found in. Because acetylcholine is really quickly inactivated by the enzyme acetylcholine esterase, so anytime you see A's at the end of a word, it's an enzyme. The effects triggered by these cholinergic neurons are quite short-lasting. So the neurons in the autonomic nervous system that produce and release norepinephrine or noradrenaline are called our adrenergic neurons. Most of our sympathetic postganglionic neurons are adrenergic. So pretty much all of them except for our sweat glands, which we mentioned earlier. Like acetylcholine, norepinephrine, which is often just represented by Ne, is stored in those synaptic vesicles and released by exocytosis. They diffuse across the synaptic cleft and they bind to the adrenergic receptors on the postsynaptic membrane, either causing excitation or inhibition of that effector cell. So adrenergic receptors can bind both norepinephrine and epinephrine. And the norepinephrine can either be released as a neurotransmitter or as a hormone. So either by a neuron or by those chromaffin cells in the adrenal medulla. There are two main types of our adrenergic receptors. These are our alpha and our beta receptors. And these are found on the effectors innervated by most of our sympathetic postganglionic axons. These receptors are then further classified into subtypes. So we have alpha 1 and alpha 1 and 2, and then beta 1, 2, and 3 receptors. So you'll see the little alpha sign with a superscript 1 or 2, or the beta sign with a superscript 1, 2, or 3. And they're classified based on the specific responses that they elicit when that norepinephrine binds to them. So although there are exceptions, generally the activation of our alpha 1 and our beta 1 receptors will produce excitation. And the activation of our alpha 2 and our beta 2 receptors will cause inhibition. So these will pass on an action potential and these will stop it. Our beta 3 receptors are only found on our brown adipose tissue or our brown fat, and their activation causes thermogenesis or heat production, so important in babies. The activity of norepinephrine at a synapse is stopped when either the neurotransmitter is taken back up by the axon that released it, or when it's enzymatically inactivated. So an enzyme comes in and inactivates that neurotransmitter. Compared to acetylcholine, norepinephrine actually lingers in the synaptic cleft for a bit more of a longer time, and thus the effects triggered by our adrenergic neurons typically last a bit longer than those triggered by our cholinergic neurons. So lastly, taking a look at all of that in the form of an image. So this first image here is looking at a sympathetic preganglionic neuron. It releases acetylcholine, so this is a cholinergic neuron. Here, the acetylcholine binds to a nicotinic receptor, which produces excitation, so the action potential running along this neuron will pass to our postganglionic neuron. Our postganglionic neuron is an adrenergic receptor because it adrenergic neuron because it releases norepinephrine. It releases the norepinephrine and that binds to our effector cell. Second, we have another sympathetic preganglionic neuron which releases acetylcholine and is therefore also a cholinergic neuron. Acetylcholine binds to our nicotinic receptor, producing excitation and passing on that action potential. The sympathetic postganglionic neuron is another cholinergic neuron because it also releases acetylcholine. And then on the surface of our sweat glands, we have muscarinic receptors, which will either depolarize or hyperpolarize, so either excite or inhibit when that acetylcholine binds. Lastly, we have a parasympathetic preganglionic neuron. So again, remember all of our preganglionic neurons are our cholinergic neurons because they release acetylcholine. Acetylcholine binds to a nicotinic receptor, passing on that action potential to our postganglionic neuron. All of our parasympathetic postganglionic neurons are also cholinergic, so they all release acetylcholine. That will then bind to the receptors. Here we have a muscarinic receptor on our effector cell. So the muscarinic receptors having quite short-lasting effects. Okay, so now we are all done and dusted with the nervous system. Bit of a shorter lecture to finish that off. Any questions you have about the entire nervous system, we can cover in the revision tutorial this week. I'm also going to post some revision questions which tie all of this information in together so you can kind of see how it all works. Thank you again for listening, and I hope you have a nice rest of your day.